Welcome to Bio 326, Human Physiology. Let's get started on some basic concepts in Chapter 1. Physiology incorporates a little anatomy, some chemistry, some physics, and a little math. Success in this course is going to require you to understand how organs and systems function on a grand or macro scale, and also how all the little pieces fit together. This means physiology is both memorization and big concept ideas. Whenever possible, attempt to explain a process in your own words like you were teaching someone else, and think about how that process would be affected if various parts were absent. Even if you know all the chemical and physical aspects, organisms are still much more complicated than the sum of their parts. This is the idea of emergent properties. These are properties that cannot be predicted by simply knowing the anatomy and function of the components. For example, we know the parts of the brain and how the nervous system works, but we still can't really fully explain properties like emotion, personality, and intelligence. And also keep in mind, although we know a lot about the human body, there's still processes we don't fully understand, and new information is learned all the time. What you learned this year may have to be modified in the future. Our text this semester really highlights the organizational levels that I put down there in the red font. Bonds between atoms form molecules, molecules form organelles and cells that are surrounded by membranes, collections of similar cells create tissues, and then we get to organs when we have several tissue types that are united to form together to form a structure with a common function. And looking at life processes, our cells really are just kind of like mini people. They do everything that we do. They have to grow and differentiate, reproduce. They have to eliminate wastes. They need to take in oxygen, energy, and nutrients. They need to move. They need to maintain their boundaries. This is all kind of the big concept of homeostasis. Your cells do everything you do and need everything you need as a fully functional human being, which is really kind of neat, I think. How many cells do we have? This is a common question that comes up. This is always up for debate, but probably about 30 to 40 trillion cells, and this is going to create a huge organizational challenge. Systems in the body are very integrated. Our text by D. Silverthorne highlights the integrative nature of physiology and makes a lot of connections between different organ systems. And on this figure, we see some of the systems that we'll explore in this course. Figure 1.2 really highlights the fact that our body consists of many integrated systems that have to interact with each other as well as the external world. Think like your lungs and your GI system. One thing that we will see with all of this integration is that problems with one organ system always create problems with at least one other organ system. And then looking at the figure above, you should be able to see which 4 organ systems actually communicate with the external environment. There are several key concepts that will feature repeatedly throughout this course. And the first is the relationship between structure and function on molecular, cellular, and even the entire organ system levels. Physiology really boils down to a gazillion, that's my scientific term, molecular interactions. And these interactions are often facilitated by proteins, especially enzymes and receptors that depend on a very precise structure for proper functioning. Enzymes function in a lock and key fashion, and if their structure changes, then the appropriate enzymatic key may no longer fit well. Structural changes can happen intentionally in the body, maybe to slow down or speed up a particular function or reaction, and this can happen as a side effect of just non-ideal conditions in the body, say stress from heat, cold, changes in pH, dehydration, etc. All of these non-ideal body conditions are stressful because they change the structure of proteins, and then those proteins are no longer efficient at doing their job. Another big theme is compartmentation, which is kind of like the division of space. This is, again, a huge organizational challenge. We need to specialize and isolate functions, especially competing ones. Say a cell that has protein synthesis occurring in one area and protein degradation in another area inside the same cell. Or maybe functions that require very different microenvironments, say like your stomach with a very acidic pH versus your blood that has a slightly basic pH. At the cellular level, compartmentation is based on the idea of membranes. So membranes equal surface area. The more membranes that you have, the more surface area cells have to work with. And this means they have more control. Compartmentation is also necessary for protection. Protection from what? Environmental changes in salinity, pH, metabolic waste accumulation, pathogens, pressure, etc. And the big thing here is fluid compartments are going to be much more important than physical or anatomical compartments. Another big theme is the requirement of energy. What do we need energy for? We need to transport materials within the body, like blood, sperm, hormones, air, food, electrical impulses. We also need to move substances across cell membranes for exchange and storage. We also need to be able to synthesize new materials. This takes energy. We also need to be able to convert materials to new products. So lots of energy is going to be required. Another theme is the idea of information flow. Information flow between cells is usually chemical or electrical in nature, but occasionally mechanical information, think like pressure, stretch, and temperature, is used. Because the body has so many compartments and within and across so many different systems, effective communication needs to occur between all of these systems to maintain proper functioning. And all of these themes together result in this grand idea of homeostasis. So if you break that down, homeo means similar and stasis actually means condition. So homeostasis is kind of like keeping a similar condition of all of these variables. Another way to consider homeostasis is that it's the dynamic vital balance. Dynamic because things are always moving. Vital just means necessary for life. And then the idea of balance means that variables should stay within an acceptable range rather than at a fixed set point. Note that homeostasis does not mean static or unchanging. Variables that are going to be regulated are things like pH, fluid volume, nutrient concentration, temperature, osmolarity, blood pressure, heart rate, etc. And you want to think about what the word pathology means. Pathology means the disruption of homeostasis. And anything that is a pathology can lead to disease and possibly death. And I want to mention the idea of the set point. That's going to come up a lot. The set point, when we talk about that, is kind of like the best optimal value for a variable. For example, we always think of the set point for body temperature being 98.6 degrees Fahrenheit, or blood pH has a set point of 7.4. While the acceptable range for these variables might be slightly above and below the set point. And another thing, homeostasis doesn't mean that nothing ever changes in the body. Actually, things are changing all the time in the body. So rather, is what we're going to see is that, homeostasis is about being able to detect changes in these variables and then trying to correct those changes and bring those variables back into that acceptable range. So it's a very dynamic process. This graph up here shows a dynamic circadian rhythm. Circadian actually just means about a day. So we have a circadian rhythm graph here for the set point of body temperature. Our body temperature lowers throughout the night and then elevates throughout the day. One reason you might feel crappy if your alarm wakes you up early in the morning is that your body has not prepared for being awake by warming up a little bit. And then also notice this drop off in set point during midday. This is often a time of day when you feel kind of sleepy. And so the exact times of these temperature cycles will be slightly different for each individual. For us as human beings, our external environment is the outside world. But for our cells, their external environment is what we call extracellular fluid. This is fluid that is outside of, but yet in direct contact with cell membranes. The salinity, waste concentration, nutrient and gas composition, pH and temperature of the extracellular fluid directly impacts the cell. So another way to think about homeostasis means that you are trying to kind of maintain a relatively constant extracellular fluid. Most of this course is going to be about what kind of variables change in the ECF and the mechanisms in place to correct these changes so that the variable in question kind of, again, moves back into that acceptable range. And I want to draw your attention up there to the blue word, lumens. A lumen is a space inside a hollow organ. So think like your hollow stomach, bladder, your intestines, your trachea. Material inside of a lumen is still considered outside of the body until it crosses a cell membrane and moves into a cell. Lumens are always lined with epithelial tissue. A little more on this later. Therefore, lumens are really part of the external environment. So anything that is within a lumen, even though it's inside your body, is really not in you. An analogy: if you have a donut and you place a straw through the donut hole, anything moving through the straw, which would be the lumen, is moving through the donut's lumen and is not part of or in direct contact with the donut itself. And then last on this slide, we see and we know that most of the body is composed of water. And if you break down this fluid, about 2/3 of our bodily fluid is in the ICF, or intracellular fluid, and only about 1/3 of our body's water is in the ECF. We can also kind of subdivide the idea of ECF. So ECF can be either plasma, so this is the fluid portion that's inside your blood vessels. And the other part of ECF is what we call interstitial fluid. This is the fluid that surrounds your cells. So homeostasis does not mean equilibrium. Instead, homeostasis is a dynamic, steady state. This means that materials are always moving. This is a dynamic process. Things are moving across cell membranes and between fluid compartments, but steady in that there is no net movement. And the idea of equilibrium means that the composition of all compartments is exactly the same. So the goal for homeostasis is to maintain this dynamic, steady state of disequilibrium and not go to equilibrium. Our normal functioning requires different concentrations of substances and ions between compartments, between organelles and cells, and much energy is used to maintain disequilibrium. For example, there's more potassium inside our cells than outside, and there's more sodium and chloride outside of our cells than inside. This is absolutely necessary. When death occurs and energy is no longer available to maintain this vital and necessary disequilibrium, our cells just naturally go to a state of equilibrium, which is bad. Homeostasis requires that our bodies and cells have control systems to detect changes and to correct them. This is kind of like the thermostat of your house that detects when the temperature gets too high or too low, and adjusts your furnace and air conditioner as appropriate to keep the temperature relatively stable. So a little bit about these control system components. We have the idea of local components and reflex components and control systems. Local control just means that there's no involvement with the central nervous system, no involvement with the brain and spinal cord. Reflex means systemic, or the whole body is being manipulated. Some examples of local control: cells sensing a decrease in oxygen level and stimulating just localized blood vessels to dilate to bring more oxygen to them. Or maybe stretching of the stomach with food ingestion will cause just those stomach cells to release more hydrochloric acid. A reflex or systemic control example: this would be like maybe when your blood pressure decreases dramatically after losing blood in a car accident, and you have this systemic and whole body response of increasing your heart rate, constricting your blood vessels, decreasing your kidneys' urine output. A bunch of organs all over the body are going to have to come together to reflexively and systemically correct a problem. We'll talk more about some more of these control system components as we go, But I want to kind of mention the idea of integrating center. So integrating centers, when you see that term, I want you to think that this is the decision maker. Sometimes integrating centers can be cells, sometimes they can be receptors, which are part of cells. Sometimes we think of the integrating center as an entire organ, like the brain. So again, just a little bit more about local control versus reflex control. Local just means the response is restricted to the area where the change took place or the stimulus was detected. We'll see a lot of examples of local control. Here's another one. This would be like when the blood vessels just in your fingers constrict to conserve heat on cold days. The 7 response loop components that I highlighted here in red, these are going to come up many times this semester, so a little bit more on these later. But the whole goal of a feedback loop, especially if it's a negative feedback loop, is to kind of stabilize the variable and again, move that variable closer to the set point. So most of the feedback loops that we're going to discuss are negative feedback, but we'll also talk about positive feedback. So negative feedback is what we consider homeostatic. This is the most common type of feedback loop. This is going to attempt to correct the stimulus and bring the changed variable kind of closer to the set point. Positive feedback, much less common. Sometimes I think about this like the snowball effect. Some outside intervention is going to need to stop the stimulus because with a positive feedback loop, the stimulus is being reinforced by the effectors. This is going to mean that the variable in question is going to move further and further away from the set point. And we'll see a few examples of positive feedback as we go. Here's an example of negative feedback being used to cool off, say, a hot person. Notice the variable is temperature, the stimulus is too hot, and the response by the effectors, which in this case would be the sweat glands, would be to sweat and cool the person down. So negative feedback means the response or effect is opposite that of the stimulus. Negative feedback negates. It's the opposite of the stimulus. And then last, positive feedback means the effect or response is actually the same as the initial stimulus. And again, it's going to reinforce it. The response from the effectors is going to increase the stimulus and push that variable even further away from the set point and further out of normal range. So using the temperature example on the previous slide, this means that if you were hot, the response instead would be to shiver, to warm you up and make you even hotter. Obviously, we don't want to try to warm up more if we're already hot. So again, positive feedback examples in the body are rare and they are not considered homeostatic, at least generally. And to stop a positive feedback cycle from just continuing and snowballing, some outside event needs to occur or the response will drive the stimulus and variable further and further and further away from the set point.